I've been coming to Fowler's Gap Field Station, which is located about 1,000 kilometres west of Sydney in the Australian Outback, for the past 11 or 12 years. And given the amount of time that I've spent out here, I think it's quite a special place for me, I guess, and I almost feel like home here. Now, the research station itself was established by the University of New South Wales in the 1960s, really to understand how sheep and the domestic animals impact the ecology of the arid zone, and also to understand really what the carrying capacity of domesticated animals is out here.
But I'm a behavioural ecologist, and that means I'm interested in studying the behaviour of animals in the context of their evolutionary past and their current ecology. By ecology, I mean the environment that you see before you, the climate that one experiences here, and also the social environment, which is the environment that the individuals of the same species experience when they interact. I'm actually based at the University of Exeter in the UK, and although I've dabbled in studying mammals, my favourite animal of choice are birds.
Now, in 2004, I established a long-term study of chestnut-crowned babblers out here. And these are starling-sized cooperative breeding birds found only in the semi-arid and arid regions of southeastern Australia. A range of factors drew me to study this species, and perhaps the most important one for me was that they'd never been studied before, and I really relished the idea of establishing my own study system, particularly in an understudied desert type environment.
I did my PhD on one of the rare cooperative breeding birds in the UK called the long-tailed tit, and I've been really fascinated by cooperative breeding ever since. Being a cooperative breeder means that you receive help with rearing your offspring by other, generally non-breeding individuals called helpers. Now, this is very exciting from an evolutionary perspective because it's not immediately clear how genes for a behaviour are passed on to following generations when individuals refrain from breeding by themselves and instead help somebody else to do so.
Now, chestnut-crowned babblers form quite large social groups which forage together throughout the day, as well as collectively roosting together at night time. A typical day in the kind of fieldwork, in our fieldwork on this system, really involves leaving the field station pre-dawn, either to establish the presence of the babblers in a given area, or to catch them using mist nets. And the advantage of being in the field this early is that babblers call very loudly to each other, actually just at dawn. And so it allows us to be quite precise with our knowledge about where the groups are at a given moment in time. And we also take advantage of their tendency to fly between areas of cover in very close proximity to each other. We then can quite easily herd the birds into a line of mist nets that we've set up the previous day.
Following capture, we take standardized measurements to assess the size and condition of each bird, and we fix a uniquely numbered metal ring and a unique combination of coloured rings around their legs for identification in the field. We also take a small blood sample to determine the gender of the birds and their levels of genetic relatedness. Finally, we insert a small microchip transponder under their skin, similar to that used in pets.
Now, the reason for using the microchip is to really take advantage of the shape of the babbler nests. Now, because these babbler nests are dome-shaped, it means the birds must pass through an entrance hole in order to feed the chicks inside. And what we can therefore do is put a copper coil antenna around the entrance of the nest, knowing the birds must pass through this antenna to feed the chicks. Now, this means that we can then record the identity of every bird that goes through that nest antenna along with its date and its time of entry, and we can therefore record with very precise detail over many, many days at a time which individuals belong to which groups, as well as which group members contribute more or less to feeding those offspring.